Hi, it's Mr. Riku, and today we're going to talk about proteins, standard level and higher level, and additional higher level. The main objective of this lesson is to examine the chemical structure of amino acids and proteins and relate to their biological function in cells and tissues. Okay? Okay, so here first we have an image of a protein. Okay, so we can see the image of glucagon. Each one of these circles represent one type of amino acid. So in this case, it's a quite small protein. It's a very small protein, but we can see how each one of these amino acids is interacting with one another and folding to form this 3D structure that is vital for the function that this protein has. Okay, before we talk about the structure of proteins, let's talk about their building blocks, the amino acids. So this is the typical structure, this is the universal structure of amino acids. We have an amino group here in the left that has a nitrogen, making two bonds with hydrogens connected to an alpha carbon in the middle, in the center. This alpha carbon has a single bond with this hydrogen that is also connected to a carboxyl group. So COOH, double bond with oxygen, and then OH, plus R group that we called variable group. This R chemical group is what determines the type of amino acid that we have um, in the molecule. We have 20 types of amino acids, so we have 20 types of R's. Of course, you don't need to remember, you don't need to memorize these variable groups. You just need to know that they are highly variable and they change in terms of charges, polarity, and affinity to different other chemicals. Okay, another view. Again, we have the amino group. We have the alpha carbon that is linked to the side chain. The side chain is this R group that is defining for the amino acid. Plus, we have the carboxyl group, making one bond with an oxygen, and then with a double bond with another oxygen. Okay, so we can represent this in a variety of forms. You can show like this two-dimensional structure formula, just showing the traces and the simple lines. You can form circles, you can differentiate by colors, but you can show like more 3D with the bonds protruding or moving backwards to the screen, but they show the same thing. They represent the same type of chemical. Again, amino group, carboxyl, and alpha carbon. This is key. This you need to know because it's the essence of all amino acids. And the amino acids are the basis of all proteins, and the proteins make up all life in this planet, including you. All right. Again, another way to represent, we have the R group, alpha, and amino. Here we have some examples of amino acids. Okay, so we have, for example, glycine, a very small one, alanine, very small amino acid. But if we check methionine or, for example, tyrosine, they have more complex R groups. They have a different chemical interaction with the chemical world around them. Okay, this is going to affect the mass of the molecule, the size, the interactions, the charges, the solubility in water, the interaction with water molecules, or also the interaction with other chemical groups. Okay, as I told you, there are 20 types of amino acids. Of course, you don't need to remember them. I don't remember them. We don't need to know exactly their names or their chemical compositions. It's too much. But what is important for you to know? That there are 20, and each one of them, they have a property. Okay, some of them are polar, non-polar. Okay, some of them are charged positively or negatively, and this is going to affect how do we build all proteins that exist in the planet, combining these 20 amino acids. Okay, depending on the sequence and depending on the size of the polypeptide chain, you're going to have millions of different types of proteins. Okay, good. How do we form a protein? Let's start with the simple. We remember when we talked about carbohydrates, monosaccharides and disaccharides, the same thing applies here. If you have one amino acid, if you combine with another amino acid, this is what we call a condensation reaction. Typically, enzymes are going to perform this. So they're going to bond this hydroxyl group with this hydrogen atom, and then you're going to produce as a product, you're going to condensate one water molecule, as we can see here. So one tiny water molecule is produced. And you have two amino acids linked that we call D2 peptide. Peptide is our amino acids being connected. Okay, so we have a peptide bond, that is this chemical covalent bond between the two amino acids, and that's it. You have a dipeptide. Good job. And you condensated one protein. Okay, if you continue to do this over and over and over and over again, maybe hundreds of times, you can fold a protein. You can make up a protein. Okay, and this protein can be can suffer denaturation, can be denatured. We're gonna see how it works in a few minutes. One interesting practice for us is to model amino acids. This is great because you can practice with the different atoms and visualize their composition and their interaction and their bonding. Okay, so this is cool. This is interesting for us. Okay, so now denatured proteins. Again, proteins are typically made of hundreds or thousands of amino acids, and they typically form a globe or 3D structure. Okay, and they need to keep this structure like a piece of plastic. It only has a function, for example, like this. Okay, this control. This has a function because it has a specific shape. If you melt this plastic, you lose the function, right? The same thing with proteins. If you change the shape, the 3D conformation, the 3D structure of a protein, you change the function. You lose the function, in other words. Okay, so again, there are 20 types of amino acids, okay? And the main factors that can affect their structure, that can denature, are pH and temperature. Okay, so pH and temperature. Why pH? Depending on the concentration of free hydrogen ions, free protons in the solution, you're going to interact with the different charges of the R groups and the other chemicals. So this can affect, can displace, can change, can affect the interaction. So different pHs are going to have different effects on proteins. So typically proteins are adapted to a specific pH. And temperature, okay, specific extreme temperatures and extreme pHs, they're going to affect. Temperatures affect because they make this so thermally dynamic that can eventually break up and disrupt the chemical system. The DNA, the nucleic acids, they code for these 20 amino acids. So we have the genetic code. We're going to cover this in the future. But the genetic code codes for the 20 amino acids. And depending, again, on your sequence and the composition, you're going to have millions of types of possible proteins. All right. Now let's talk about the structure of proteins. The first structure we're going to see is the lineage, is the line of amino acids, the sequence of amino acids. As you combine the sequence of amino acids with one another, you form a secondary structure. Okay, when you fold the line of amino acids on one another, you can have two main types: the alpha helixes that are stabilized by hydrogen bonds, or the beta pleated sheets that you form these barrels, these parallel structures, and they're also stabilized by hydrogen bonds. And as you make up the protein, primary, secondary, tertiary, or even quaternary, you make up a 3D shape, a globular protein, as they're typically folding in. All right. And this, again, this structure of this protein that is key, that is vital for its function. If you increase or decrease the pH, or if you denature by crossing, for example, the 40 degrees Celsius boundary, you can denature and you can lose the function. This is why fever is dangerous, because extreme fever can really harm your body. And this is why you can never put your hand on a hot plate, because you basically destroy your proteins. You basically kill your proteins in your cells, and then your body creates a sensation of pain to warn you to stop doing that. Okay, this is the cell level. The cell level covers the chemical structure, the key chemical structure of proteins, and the additional high level is going to focus on the types of structures, the conformations, the levels of organization inside polypeptides. This is the additional high level for B1.2. So again, the DNA codes for all these amino acids, all these 20 amino acids. All right, and we have 4 types of structures of proteins. We have primary structure, secondary structure, tertiary, and quaternary. We're going to see exactly how each of one of them folds up. Okay, so first, the primary, very simple, straightforward. It's the line of amino acids. Okay, is one connected to the other. Each circle here is representing one amino acid. As you link one to one another, that's it. You have the primary structure. Secondary structure, when you fold the primary around itself. You see here in green, the amino acids, they're linked and they're interacting with one another. You can form the alpha helixes or the beta pleated sheets. The beta pleated sheets are parallel strands that pair. Okay. What is going to dictate each one of these sequences in different proteins? The sequence of DNA and the sequence of RNA and the sequence of amino acids. Depending on the sequence, you have a different composition, you have a different secondary structure. Next, we have tertiary structure. How do you make a tertiary structure? Combining secondary structures. Eventually, these secondary structures, here the blue line, they're going to link, forming hydrophobic interactions, hydrogen bonds, disulfide bridges, or even ionic bonds, attracting or repelling the secondary structures. These chemical attractions or chemical interactions might conform, might make a different form in the protein. This is a tertiary. All right. The major part of proteins stop on the tertiary. We can even have a quaternary structure. What is a quaternary structure for protein? When you get more than one tertiary structure and you combine. The most beautiful example is the hemoglobin. So in the hemoglobin, you have alpha and beta units, tertiary structures that you link, that you combine, forming this complex quaternary structure. And we even have the heme group, the heme groups containing iron in the center of the molecule that attracts oxygen and can move through the body, deliver oxygen through the body. So this is one example of quaternary protein. Okay, and these quaternary proteins and other proteins as well, they can be conjugated or non-conjugated. Non-conjugated, simple, only amino acids. Conjugated because they are conjugated with another chemical group, another extra group, for example, carbohydrate, as the proteins in the glycocalyx and membranes, or a metal ion like iron in the hemoglobin. So hemoglobin is a conjugated protein. The major part of proteins, they form a globular shape. Okay, they have a globular shape. They make a conformation, a 3D conformation as a globe. All right. But we have several proteins that have a fibrous shape, a linear shape. This is the case of collagen. So we see that the amino acids are interacting in a way that they form a line of amino acids. Okay, this is also another 2 types of proteins. And in the end, the fibers or the globular proteins, they can be denatured. And if they are denatured, they lose their function. Again, if you change the form, you change the function. But when you change the function, typically you lose the function. That's it for today. I hope that was helpful, and thank you for visiting.